Hi, welcome to the lecture on EnvAdapt48 in animals. Now we will focus on the muscle structure, because beside the energetics and beside the freeze tolerance or freeze avoidance and etc., animals usually are moving. And we have discussed a lot of running and escaping during this course. And so far we have discussed that, okay, the prey can run and escape if it's able to produce enough ATP for the muscles. And for that it has three different ways. It can increase the anaerobic catabolism, it can have oxygen store, it can have phosphate store. And if it can produce enough ATP, then no problem. But the problem is that, okay, you also need these muscle cells, muscle fibers to contract. And you can have different kind of fibers that are contracting fast or slow, and some of them can contract only for a short period. The muscle overall structure is something that you all know. We have muscle tissue that is consisting of these contractile cells, and the movement is based on two proteins, a molecular motor myosin, and that is interacting with another protein actin. And they are producing mechanical force from the chemical energy of ATP. And so far we have learned that, okay, when turning this ATP to mechanical force, you are losing a lot of energy as heat. And we can divide muscles easily on the striated and smooth muscle cells. The striated muscles have transverse bands, as in the skeletal muscle picture made with a microscope. So there are bands that are in the same direction. And there are regularly repeating units called sarcomeres. And in the smooth muscles, there are also myosin and actin, but not in this kind of structure. So there are no sarcomeres. So they are more in the random directions. Let's focus on the fine structure of the skeletal muscle. Skeletal muscle cells, skeletal fibers, are quite extraordinary cells in the animal body. The length can be about or even 30 centimeters. And they are not emerging in your body with the same typical cells are dividing and then modulated and regulated to form different kind of cell types. But now these are formed by merging hundreds of myoblast cells together. So that's why they can have hundreds of nucleus. And that makes them able that, okay, even these maybe 100 micrometer long cells can, when they are merging together, you can make cells that are 30 centimeters long. But still, although they are very long, they are still keeping quite thin. So the diameter is perhaps 10 micrometer or 100 micrometer. And the extreme wide skeletal muscle cells can be found, surprise, surprise, in the Antarctic fish, several hundreds of micrometers. So, and this thickness is to be a thin cell, it's very important for this regulation of contraction. But we will discuss this a little bit later. And if we look a little bit more closer on this structure of these muscle fibers, muscle cells, there are different areas. So they have these myofibrils that are having, the diameter is about 1 or 2 micrometer. And these myofibrils are there side by side, and they have repeated transfer bands, A band and I band, and then there is a jet disc in the middle of each I band. And because when you are staining your microscope preparations, during the microscope preparations of a skeletal muscle, these A bands and I bands, they are having different colors. So that's why you can have the striated structure. But let's focus more detail on the sarcomere structure. So we have thin filament and thin filament. The thin filament is caused by actin, and not one actin, but several actins. So as actins are binded together to form this kind of macroscopic structure. And beside the actin, there is also troponin and tropomyosin. And then this thick filament is based on the myosin, and it looks completely different. A single myosin looks a little bit like a snake. So there is a tail of myosin heavy chain, and then there is a light chain of the myosin with a hinge, and then the head structure. And this head structure can turn and make cross bridges with the actin. But beside these actin and myosin, there exists also titin or titin. And this titin is attached on both M line, so in the middle of the thick filament, and Z disc, so that is in the other end of the thin filament. So it goes all the way, but it's quite flexible in the I band, so it has some kind of increasing the somehow it's supporting this sarcomere structure. Then beside titin, there is inelastic nebulin that is stabilizing this thin filament, so keeping it straight and keeping the structure. So there are some supportive proteins, and then in the thin filament itself, we have troponin and tropomyosin, and they are very important for this cross-bridge interaction. So they are regulators of this whole contraction. And if you take a cross-section of this sarcomer, you can see that, okay, what exists there in the I band and A band. So in the A band, there are two kinds of particles. So there are thick and thin filaments. Then you have areas where you have only the thin filaments. Then you have areas where you have only thick filaments. So these small particles cannot be seen. So that makes it sense that, okay, now if you are moving these filaments, then you are maybe affecting that, okay, how wide these zones are. And from that, they already got in the 50s on the sliding filament theory. So if you have an electron microscope picture of the filament, you can see that in the center it's completely black. And on the left hand and right hand side, it's quite pale. So there are areas where there is only thick filament, areas where there is only thin filament. And if you are contracting, you are getting, for example, this H band and I band less thin. So so it's more narrow until when the cell is fully contracted. Now these thick and thin filaments are completely overlapping, and you can't anymore see areas where there is only thick or thin filament. So the sliding affects that, okay, now these H and I bands are more narrow because they are overlapping. And this length of these filaments are not changed. So they are just overlapping. So they are just moving across each other. And actually, how much they can move is affecting on how much they can make force. So in the 60s, someone was making a quite nice experiment on the skeletal muscle that, okay, measuring that how long is the myosin, how long is the actin, and what is the length of this whole filament. And then how much force, what is the tension of the sarcomere. And when they are more closer to each other, they are making more force, so there's tension that can be produced. But sooner or later, the thick filaments are touching each other. And if they are more overlapping, then these thick filaments are overlapping, and that will reduce the force that they will make. Until, in the end, the tension is actually very minimum when the cell is fully contracted. So, in that case, the length of the sarcomere is exactly the length of the thin filament. Okay, let's have a closer view on how the myosin and actin are interacting. So we have myosin, we have actin, and they have binding site for each other, and the myosin has also binding site for ATP. And the structure of myosin when it's binding on actin is just a transient state. So if you have any mechanisms to remove the myosin from the actin, the myosin structure will be changed. And how that can be regulated is by binding ATP. So that dissociates the myosin from the actin, and then the myosin goes in more stable conformation. But at the same time, this myosin ATPase activity will hydrolyze ATP, and this energy is used to turn again this head group. And if there is calcium available, this cross bridge will be formed. After which the phosphate will be released and the filament is moved by 10 nanometers. And after that, the ADP is also unbinded. And then you can have the second round and second round and second round. And so it seems to be that it's you need ATP for the contraction, but you also need the calcium. Let's look a little bit more detail. What is the role of tropomyosin? What is the role of troponin? So when there's no calcium, the head group is touching only loosely on the thin filament. But there's tropomyosin that is blocking this myosin binding site. But then, if you have calcium, the calcium is binding on the troponin, and this will turn the structure. So the myosin binding site is exposed. And then this myosin head binds more tightly. And this causes that then the thin filament can be moved.